Hello all. Our today's topic is Ebola virus, part 1. This is from chapter Viral Diseases from Clinical Microbiology, MB-703. Here we are going to study viral diseases, in that the disease caused by Ebola virus, which is causative agent of Ebola, general characteristics of the Ebola viruses. It is also called as a Ebola virus disease, EVD. It is described as one of the world's most virulent diseases by World Health Organization and was popularly known as Ebola hemorrhagic fever in the past. It is a highly contagious disease. It is usually considered a severe and deadly illness. Outbreak of Ebola virus disease have shown to have a very high fatality rate, ranging from 50 to 90%. That is, the mortality rate, if you consider with other viral diseases, it is too high. Ebola virus was named after one river called as Ebola. This one from northwestern Zaire, now the Democratic Republic of Congo, where the first documented outbreak emerged in 1976. The virus is transmitted to people from wild animals and within the human community through human-to-human contact. That is, this is a zoonotic type of a disease. Natural host for Ebola virus is not yet conclusively identified, but the most probable host appears to be the fruit bats. These fruit bats are from Pteropodi family. Ebola viruses are highly infectious in nature and were once the sole reason for contraction of this disease by a scientist while conducting an experiment in the laboratory. That is, you can say this one is a nosocomial infection, hospital-borne infection, but mainly observed in hospital staff workers. There are 5 species of Ebola are known, all named after the region where has been identified. These species are Zaire species, Bundibugo species, Sudan species, Reston, and Thai forest species. About the Zaire species, here the fatality rate, mortality rate is observed up to 80%. There is range of mortality rate and it's up to 80%. While in case of a second species, that is Bundibugo species, mortality rate is observed up to 40%. Third one, the Sudan species, where it was causative agent of some past outbreaks. In case of a Reston species, no human cases reported, while in case of a Thai forest species, it was discovered among 2 wild chimpanzees. Species greatly differ in the characteristics and fatality, but most severe infections, they were by Zaire species from South Africa. About the virus, this virus is belonged to the family Filoviridae. The virus is non-segmented filovirus, membrane enveloped filamentous viruses that contain a negative sense single-stranded RNA. The long filaments are 80 nm in diameter and either 800 to 1000 nanometers long. And RNA is only 1% of the mass of the virus. In this picture, you can see the viruses which are looking like number 6. The shape of this virus is variable. That is, some may show long tubular structures like this, while some may show branch structures like this. The viral particles look like long stretched filaments with some particles tending to curve. Okay, here you can see there are number of curves may present in this virus particles. So they tending to curve into an appearance like the number 6. Here also that 6 shaped virus, Ebola virus, and it is primarily composed of 3 compartments. These 3 compartments are the nucleocapsid. See here, nucleocapsid. This one is a nucleocapsid. The matrix space. This one is a matrix space. And lastly, the envelope. Okay, this one is the viral envelope. So this is composed of these 3 main compartments. With that, other proteins also present, in that polymerase and glycoproteins, as well as VP24. Okay, this protein is important. This nucleocapsid of the virus is composed of nucleoprotein. Okay, this one is composed of nucleoprotein NP, VP30, VP35, housing this RNA genome of Ebola virus. The viral envelope is derived from the cell membrane of the host during the budding process. Transmembrane glycoprotein, these are present on the inner lipid bilayer surface as 7 to 10 nm long spikes and play significant role in Ebola virus life cycle. They mediate attachment of the virus as well as their entry and fusion into target cells, that is, to the cells where they infect, and thus permitting replication as well as spread of the virus. The matrix space of the virus is between the virion envelope and central nucleocapsid houses viral matrix protein VP40 and VP24. This VP40 mediates budding as well as a viral particle release. So their role is very important in the pathogenesis at the same time while preventing infection. While VP24, a minor matrix protein, it participates in nucleocapsid formation as well as assembly, also regulating viral transcription and replication. In the center of the virion, as you know, a nucleocapsid is present, which is composed of series of viral proteins. These are attached to linear non-segmented single negative stranded RNA genome. There are total 4 nucleocapsid proteins, and they are nucleoprotein NP, RNA polymerase, polymerase cofactor VP35, and transcription factor VP30. These are important for replication and transcription of viral genome. Besides this, 3 more proteins are present. Here, this Ebola virus is composed of only 7 genes and it coding for total 8 proteins. The 7 genes are, as you know, nucleoprotein, the viral protein VP24, VP30, VP35, and VP40 with polymerases as well as glycoproteins. In this way, these are composed of these 7 genes. Now, about transmission of Ebola viruses. The exact natural reservoir host for Ebola virus still remain uncertain. On the basis of available evidences and nature of similar viruses, researchers believe that This virus is zoonotic, that is, animal borne, and is totally dependent on its host for replication as well as overall survival of the viruses in the body of the host. Concurrent transmission of Ebola virus disease occurs through two main routes: animal to human and human to human contacts. Here, nowadays, this one is considered important, human to human contact, because of which spread of Ebola virus disease once it enter from animals into the human. Animal to human transmission occurs through handling bushmeat. This bushmeat means meat of wild animals, or because of a direct contact with infected non-human primates such as chimpanzees, gorillas, fruit bats, as well as monkeys. In Africa, there is a practice of eating bushmeat, that is, meat of all these animals, because of which possibility of a transmission of infection from animals to human. Human to human transmission is also one important route in the transmission of Ebola viruses. It occurs through intimate contact with infected cases. Exposure to the blood and other body fluids of patients exhibiting clinical manifestation is considered as major portal of entry of the viruses in the healthy person. Even after handling dead bodies, there are evidences showing occurrence of infection in the handlers. Blood, semen, and vomiters are most infectious body fluids. During handling, all these specimen possibility of, despite its detection in breast milk, breastfeeding is not confirmed route of Ebola virus transmission and is same applicable for droplet, that is airborne infection, as well as a mosquito-borne infection and by insect bites. That is, okay, exact route is not known if you consider droplets, mosquito bite, as well as an insect bite. About entry of Ebola viruses, as you know, after handling the dead bodies or clinical samples of the patients, there is an entry of the viruses in the human body. They are either via mucosal surfaces, abrasions, that is cuts if present on the skin, and injuries in the skin, or by direct parenteral transmission, that is possibility of accidental needle injuries. Ebola viruses then attack many other organs. In fact, the virus is able to invade almost all human cells using different attachment mechanisms for each cell type, except for lymphocytes. This is very unique type of pathogenesis. It has been proposed that Ebola viruses can enter the target cells by using different uptake mechanisms. They either use liquid draft, receptor-mediated endocytosis. This is mainly applicable for the coronaviruses, and macropinocytosis. By any one of mode from this, they may enter in the affected body part, that is in the human cells. Now I'll see in detail life cycle of Ebola viruses. It begins with binding and attachment of Ebola viruses to specific surface receptors on target cell membrane, and this one mediated by glycoprotein subunits. The virion envelope subsequently fuses with the cellular membrane of host cells, and the virus nucleocapsid is released into the cytosol, resulting in viral uptake. That is, the common mechanism of entry of viruses upon infecting target cell by recognizing receptors, etc. The internalized nucleocapsid serve as a template for viral transcription and replication, and this results in copying into full-length positive strand RNA antigenome. These antigenomes serve as a templates for transcription and negative strand virus progeny genome copies. This newly synthesized structural proteins and genomes then self-assemble and accumulate under cell membrane sites from where the virus is released. That is the common mechanism of release of viruses. When budding off from the cell, virions gain envelope from the cellular membrane and they bud off. These mature progeny particles infect other neighboring cells to repeat virion cycle. And in this way, you can see destruction of a large number of cells in the body. Replication of viruses at an extraordinarily high rate and exploiting cellular machinery by overpowering protein synthesis apparatus of infected cells as well as a host immune defenses. As you know, viruses require protein synthesizing machinery of the target cell, of the cell, host cell. At the same time, they utilizes genetic machinery of their own for proliferation into the cells. The common host targets are divided in two categories: primary host targets and secondary targets. In the primary host targets, monocytes, these are monocytes, macrophages, as well as the dendritic cells. These are considered important. And here, majority of the times, the organs like liver and adrenals, they are targeted by the pathogen, by the viruses. Secondary targets are fibroblast and endothelial cells. Here, transmembrane glycoproteins form trimeric complex that binds the virus preferentially in the earlier stages of infection to cells of mononuclear phagocytic system, that is monocytes and macrophages, and this causes cell damage. Because of cell damage, there is a release of cytokines. There are various types of cytokines. They are released upon infection to these macrophages: TNF-alpha, IL-6, IL-8, macrophage chemotactic protein MCP1, as well as a nitric oxide, they are released at this stage. And these are responsible for increase in temperature. That is, they contributing towards fever and exaggerated inflammatory responses. So here in summary, we'll see again how Ebola viruses enter in the body, why they are lethal in nature, and how this is a rarest type of disease studied before. On initial infection, Ebola viruses invades through breaks in the skin, causing cells such as monocytes, macrophages, and dendritic cells to modulate their genes. They undergo apoptosis, where it means cell death. Release of virus particles to extracellular tissues lead to destruction of neighboring cells. Viral particles migrate from the infected cells to the regional lymph nodes, causing lymphoadenopathy. Bloodstream dissemination to the liver, that is, upon entry of viruses into the bloodstream and entry of this blood into the liver as well as the spleen, induces an active inflammatory response associated with hepatocellular necrosis and lymphopenia. Ebola virus then induces release of pro-inflammatory mediators such as the cytokines. As you know, cytokines, they are mainly responsible for febrile illnesses as well as inflammation. This causes dysregulation of the immune system. This release cytokines and tumor necrosis factor alpha from infected macrophages disturb the vasculature system integrity, resulting in disseminated intravascular coagulation, that is DIC, and this results in multiple organ failure. So reference for this topic, it is shown here. You can go through this. In the next video, we'll see Ebola part 2. In that, we'll study not only pathogenesis, with that symptoms, preventive measures, as well as a laboratory diagnosis and treatment. See you in the next video. Thank you. Thank you very much.